So good morning and welcome to Madison. On behalf of the Scientific Planning Committee for this meeting, I just want to let you know this is a fantastic city and I hope you get a chance to get out and see the town a little bit. Don't miss any sessions, but go see the town a little bit. We always hope these meetings are an opportunity for you to make new friends and hopefully get recharged scientifically and learn some new science. So my name is Matthew Church. I'm at the Flathead Lake Biological Station at the University of Montana, and it's my pleasure to introduce today's plenary speaker, Dr. Susie Wood. Dr. Wood is a senior scientist at the Cawthorne Institute in Nelson, New Zealand. She's an expert in freshwater cyanobacteria with a particular focus on improving our understanding of both the mechanisms and pathways that underlie the distributions of cyanobacteria and cyanotoxins in lakes and rivers. Her work relies on a variety of approaches, including some molecular biological approaches, for example, tools that leverage the polymerase chain reaction to quantify distributions and abundances of both toxin and non-toxin producing cyanobacteria. Dr. Wood attended Victoria University in Wellington for both her undergraduate and graduate education, having received her PhD in biology in 2006. She was then awarded a Foundation for Research, Science and Technology postdoctoral fellowship from Cawthorne and University of Waikato, where she went on to become a senior scientist in 2014 at the Cawthorne Institute. She has a strong and long-standing interest in improving understanding of environmental history and water quality in New Zealand. And most recently, this includes her leadership of the Lakes 380 project, which we're going to hear more about today, which is a $12 million program aimed at building knowledge about the health and history of New Zealand lakes through an integration of water quality measurements, eDNA sampling of sediment cores, and use of indigenous knowledge. Dr. Wood has received several awards and recognitions for her science that include the receipt of the New Zealand Freshwater Sciences Society Medal in 2019 for her outstanding contributions to freshwater science and management and her leadership of women in science. More recently, in 2022, she was the recipient of the Kilham Lecture Award from the International Limnology Society. So needless to say, we're thrilled to have Dr. Wood join us to open the plenary sessions at the 2024 ASLO meeting. So please join me in welcoming Dr. Susie Wood.
Tēnā koutou katoa. Thank you, Matthew, for the wonderful introduction and welcome everyone. It's a real privilege to be here, and I'm really excited to be here for the week and learn from you all. It's it looks like an amazing program. So as Matthew said, I'm Susie. I'm a freshwater scientist, molecular ecologist. I'm super passionate about understanding, protecting, revitalising the freshwaters of Aotearoa, New Zealand, and in particular I really love working on lakes. And so it's my privilege to be able to talk to you a little bit about the project that we've been working on for the last five to 6 years. And so I'm going to start with probably the most important slide of the presentation and a little quote to get us going. So sometimes the most extraordinary things are achievable simply by doing them with the right people. So I want to start by acknowledging the Lakes 380 team. They're a group of incredibly humble and talented researchers, and the work that I present today is really the accumulation of their many hundreds or thousands of hours of work. So I'm going to start with a little bit of an introduction into New Zealand, or Aotearoa, New Zealand. So we're a small island nation nestled down in the Pacific, and we've been isolated from all other large land masses for around about 80 million years. And because of that, we've developed some quite unique flora and fauna, and perhaps the most well known to most of you will be our large flightless birds. We're also we were the last large land mass to be settled by humans. So around about 750 years ago, Polynesians and Maori travelled across the Pacific and settled in our country. And because of that relatively short period of human occupation, we have a really unique opportunity to look at the full spectrum of human impact on our freshwaters. And so we're going to delve into that a little bit today. And of course we have a set of amazing lakes, and that's what I'm here to talk to you about. So a little introduction into our lakes. We have 3,800 lakes or thereabouts that are larger than 1 hectare in size. And the blue dots on this map here show the location of each of those. And these lakes are incredibly diverse in every way, from our small urban lakes to our large lowland lakes and highly modified agricultural catchments, to our absolute gems of alpine tarns nestled up in the mountains. And they've been created by volcanic eruptions, landslides, glacial retreats, and much, much more. So you'd think with such a unique set of lakes in such a small geographic area, that would have an equally enriched scientific knowledge. But what we see, what we know is that we have scientific knowledge for less than 3% of our lakes, and those orange dots on the map show the location of those. And these monitoring records are generally pretty short, so maybe 1 to 2 decades at best. And so we know nothing about what our lakes were like prior to human arrival and subsequent human change. And of course, this is really challenging in the current environment when we think about how we're going to go about restoration and revitalisation and trying to set informed restoration targets. Okay, so most of those lakes are also our lakes that are used a lot for recreation, so they're in lowland modified catchments, and so they're not representative of all those different lake types that I introduced you to. And there's vast regions of the country where we know nothing about our lakes, such as down here in Fiordland National Park, over 900 alpine lakes that we have almost no scientific data on. Okay, so what about the current health of our lakes? So I showed you some really nice, pretty videos and pictures there at the start. So this is some results from our recent modelling work. And the different colours there represent the different trophic classes of our lakes. And what we see quite worryingly, and probably not too different from the rest of the world, is that around about 45% of our lakes are in eutrophic or worse condition. And if we focus in on Te Iko Maui, or the North Island, actually that's much worse. 80% of our lakes are in eutrophic or worse condition. And so we have like much of the world, we have a real freshwater crisis on our hands, particularly with our lakes. So this lack of scientific information on our lakes and the current poor condition of our lakes prompted a program known as Our Lakes Health, Past, Present and Future. And it also became known as Lakes 380. Probably somewhat naively, we thought we would set out to sample around about 10% of our lakes across the country. And this turned out to be incredibly ambitious, both scientifically and logistically. And so what was really important about that sampling campaign is that we set out to sample lakes across a whole range of different gradients. So from large lakes, small lakes, lakes across different trophic scales, alpine lakes. And the orange dots on that map show the location of the lakes we did sample. And as well as characterising biophysical research, we also set out in certain regions to understand more about the social and cultural history of our lakes. So just to acknowledge the different organisations that were involved, and also my co-leader, Dr. Marcus van der Goes, on this program. So this little video just shows what a typical sampling at a lake. So we're taking surface sediment, surface water samples, taking a suite of measurements to understand more about the current health and biodiversity of our lakes. And then we're also taking sediment cores. So as you all know, year by year, sediment is laid down in a lake. And we can analyse those different layers, and they act a little bit like pages of a history book, if you like, and we can understand how and why and if our lakes have changed. And so the Lakes 380 project is finished now. We ended up sampling around about 315 lakes. We generated literally hundreds of thousands of samples, and they've been analysed across the country and actually across the globe. We also undertook interviews and filming, and we'll come back to a little bit of that later on in the talk. And we've produced many hundreds of outputs, which you can see more of on our website. So today I can't possibly cover the whole Lakes 380 project, so I've just picked three different areas to give you a bit of an insight into the type of work that we did, or still doing really. So the first area I want to focus on is how do we go about with some of the amazing data that we can generate to allow us to reconstruct an entire lake ecosystem, both the landscape and what's happening within the lake. And I'm going to focus on some of our very cool little native fish in that case study. Then we're going to zoom back out and look at some of the national scale pictures, so how are lakes changing nationally, the different alpine lakes, lowland lakes, what does it mean when we bring all of that data together? And so you're going to be feeling a little bit gloomy by that point of the talk, I think. And so we're going to focus, hopefully, on a more positive note in some of the really amazing work we've done partnering with our Indigenous communities. So before I delve too much into detail, I'm just going to give you a super quick environmental history of Aotearoa. So we're going to travel back in time, and a reminder that humans only arrived in our country about 750 years ago. So prior to that, around about 85% of our country was covered in native forests, so podocarp, beech forests. Those areas that haven't got native forest are generally alpine or volcanic in nature. So 750 years ago, Maori arrived and settled, and both intentionally and non-intentionally, they burnt large regions of the forest. And so by the time Europeans arrived, probably about 50% of our country, or just over 50% of our country, was forested. Okay, as Europeans arrived, we kept on burning, we started logging, we built houses and cities, we started agriculture, forestry, we intensified our agriculture, and our lakes and rivers weren't enough like Europe, so we introduced perch and rudd and tench and so on. And so if you look at that, and if we think about the lifespan of a lake, tens of thousands of years, in 200 years we introduced a huge number of stresses on our lakes. So I'm going to come back and refer to those different periods of human occupation, pre-human, Maori and European, quite a bit through the next few slides. So if you can keep that slide and those terms in your mind, it will make more sense to you. Okay, so I'm just going to very briefly outline the methods that I'm going to show to you. So this is all around the sediment core work that we did. We're going to look at chronology. We're going to see some pollen data, which we use to understand the changes in the terrestrial and aquatic vegetation. We're going to look at some eye tracks and trace metal data, which we use to understand changes in the catchment and within the lake. Some hyperspectral data, which we use as a proxy for chlorophyll A. And then probably a lot of the data is based on environmental DNA. And this is a bit of still a new field in the world of paleolimnology, but it's a real game changer. So we can start to look at soft-bodied organisms that don't leave fossil remains in our sediment cores. So I'm going to show you a fair bit of that data. Okay, so the case study that I wanted to show to you is a little lake known as Lake Waiho. It's in the North Island. It's a lake that's nestled in a highly intensified agricultural region. And we're going to focus on our native fish communities in this lake and try and understand how and why they've changed. Okay, so I'm just going to orientate you. All of the data that I'm going to show you is going to be on a horizontal scale. So we've got the sediment core, and in this study we reconstructed 1,400 years of time. So we're going from that pre-human through that Maori to European as you go across the page there. And to start with, I'm going to show you the fish DNA data. So I don't know if any of you out there are working on contemporary fish eDNA, but if you are, you'll know that particularly in lakes, it's incredibly spatially heterogeneous, and it's actually pretty tricky. So you want to times that by 100-fold, really, when you start to work on fish DNA in a sediment core. It's an incredibly challenging thing to do, and this took us a long time to achieve it, but we're really excited to have this now over 1,000-year record of fish communities in our lake. And so I'm going to talk about focus on two different fish species, one that's disturbance sensitive and one that's disturbance tolerant. So here we're looking at the community composition. This is generated using DNA metabarcoding. And what we did find was that in the pre-human period, we detected 5 different fish. And so that's probably 5 different fish species, not very exciting for many of you, I'm sure, but that's actually fairly normal for New Zealand. That's a good diversity. And so that was around about the kind of fish diversity that we expected to see in our lake. Okay, so we've got our common bully there, which is our disturbance tolerant species, and our banded cockapoo, which is our disturbance sensitive species. It's a little bit hard to tell from the graph there, but the banded cockapoo is a pretty much continuous detection of its DNA through to that period when Maori settlement, and then it almost completely disappears from the record except for one detection. So we're interested in why that happened, why that species disappeared. So another technique that we used on this sediment core was what we call droplet digital PCR. It's not truly quantitative, but semi-quantitative, so it gives us a little bit more information on the abundance, the fish abundance across time. And that's the line that I've overlaid there. And so it was reasonably, there's some variation, but it's reasonably consistent between the pre-human and the Maori period. But what we see is it drops off about the same time as the metabarcoding data drops off for that common bullies, about 1950. So interested in what happened there and what might be some of the drivers for the loss of that species. Okay, so to start to answer these questions, we start to reconstruct the land, the changes in the land around the lake and also within the lake. So the first data I'm showing you here is the pollen, and we actually also used eDNA, but I'm just going to focus on the pollen data. And this really catchment follows that sort of almost that same trend I showed you nationally. So before humans arrived, this lake was surrounded by native forest. As Maori settled in the region, there was a lot of burning, and you see that by the bottom panel there where you see the increase in charcoal. And as Europeans settled, there's an increase in grasses and more recently the arrival of non-native trees, things like pine and willow and alder. Okay, so we can see some pretty big landscape changes that happen around the lake. So now we're just going to focus in primarily, just keep your eye on that European period, and we're looking at the conversion to agriculture. So Europeans arrived around about 1850 in this region, and from about 1900s onwards we start to see agriculture beginning. And in this case we've used again environmental DNA to look for the presence of sheep and cow DNA in the core. We also used our droplet digital PCR method to look at a bacteria that's specific to ruminants. And so we're using this as a bit of a proxy for stocking rate. So we know that post-1950 we really started to increase the stocking rate around this lake. Okay, we put more techniques on the sediment core. What we're looking here is the titanium to incoherence ratio, which is generated on the iTrax, and it's a proxy for erosion. So ignore those peaks, we'll come back to them a little bit later on, but hopefully what you can see there is post-1950s we see quite a dramatic increase in terrestrial erosion, terrestrial sediment entering the lake. And the final piece of evidence around what's happening around the lake comes from cadmium. So our superphosphate fertiliser in New Zealand is really high in cadmium. So we see that post-1950s we really started to throw superphosphate on our land around the lakes. So real intensification post-1950s. So what impact does this have on the lake? So these two lines we're looking at, the first one is the chlorophyll A. What we see is it's very consistent pre-human. Starts to be a little bit more variable and slightly elevated in the Maori period, but really jumps up again that post-1950s, so we're starting to get a really super eutrophic system. We use magnesium and iron as a proxy for anoxia, and it's sort of correlating, I guess, with the start of algal blooms. We start to see this lake become anoxic. So we can see a lake that's, we've got a really nice 1400-year record now of this lake moving from a clear water phase to a eutrophic lake. So what impact does that have on some of the bottom-up drivers which might be affecting the fish communities? Okay, so this data is again generated using environmental DNA, and we're going to look at the lines there go zooplankton, phytoplankton, macrophytes. So this is multivariant data, and we've fitted a principal response curve to that, and that's what's plotted here. And those red horizontal lines are key change points in the data. And so there are some changes in these communities, post-Maori settlement, by far the most dramatic changes occur actually post-1950. But really amazing to be able to reconstruct these entire food webs. Okay, so a lot of statistical analysis went on behind the scenes. I'm not going to delve into that too much, but we're just really going to come back and bring that back together and think about what that means in terms of understanding the fish communities. So the banded cockapoo, this is a disturbance-sensitive species. We saw its disappearance with that early vegetation clearance. And actually I should have said, as the trees were removed from that lake, it let more light into the lake, and we also saw a massive expansion of the wetlands around that lake. And so we think that this change in habitat was one of the key drivers of the change, of the loss of this species. They're generalist feeders, and so while there were a few changes in those, some of those bottom-up drivers, we don't think that was responsible. We don't quite understand the mechanisms, internal mechanisms, but we think it's mostly those external drivers that cause the loss of the species. Okay, if we look at our common bully, our disturbance-tolerant species, we see it was, as we expected initially, very resilient to change, but it really could no longer handle all of the intensification, those algal blooms, those oxic conditions. And so again, we think it's the external drivers, but it's very hard. Of course, there were big changes in our food web at the same time, so it's very hard to delineate those two different drivers at this point. So just an amazing example of how we can bring these different data sets together and start to look at 1,000 years, these really long time records, which are incredibly valuable. And if we think about restoring and revitalising this lake, we now know what fish species we might want to return to the lake, but we also know all of those other parts of the food web and just how they've disappeared, and maybe when we start to revitalise, just actually what we need to reintroduce. And once you start to generate these data sets, it's also an amazing opportunity to answer all sorts of questions. So perhaps the eagle-eyed amongst you might have noticed those grey bands in the sediment core. This is an incredibly erodible catchment, and these are actually large storm events, cyclones that come through the region. And so they put these pulses of sediment, and that was also shown in some of the earlier iTrax data. So now we've got an amazing opportunity to understand how long biological communities take to recover from storm events under different land use strategies. So that's still work in progress, but yeah, just to highlight the kind of opportunities that we have with these data sets. So this is just one example, and what we have is now many hundreds of examples like this from across Aotearoa. And it's what we see is really that every lake has its own unique story, and that there's huge value in understanding those unique stories as we think about how to go forward and manage our lakes. Okay, so that was one case study. What happens when we bring all of the data sets together? So our alpine lakes, our lowland, highly modified lakes, what does the data at a national scale look like? And so this data I'm going to show you is data from, I think it's around about 130 sediment cores across Aotearoa. And I'm just going to show you one piece of data, and this is the bacterial eDNA data. So our work has shown that bacteria are often some of the most, they respond very quickly to environmental change, and they're often early warning indicators. So in our data sets, we often see these change just before many of our more traditional paleo proxies, such as diatoms, cladocerans, etc. Okay, so this data that I'm going to show you, 130 cores, and I've cut it off at 2,000 years. So you're seeing from 2000 through to current day. And I'm just showing you a principle, or a PCOA. Many of you will be familiar with those. For those of you who are less familiar, each of those dots represents a bacterial community in one of the layers of the sediment core. The closer the dots are to each other, the more similar the bacterial communities. So I wanted to start by focusing on the axis 2 there, the vertical axis. And sorry, I should have said that the different coloured dots there represent those different human occupation periods. And so looking at axis 2, what we see is in that Maori and pre-human, we see a widespread of a very diverse bacterial community, which is what we'd expect, right? We've got a huge diversity of lakes, we should have a huge diversity of different bacteria. But as we move into that European period, what you see is a loss of species diversity, a homogenisation of the community. And of course, this is incredibly worrying. We know that as communities become more homogenous, they often lose resilience and resistance. And if we think about what's coming in the future, we know that in places like New Zealand, more storm events, continued anthropogenic actions. Okay, if we look at the axis 1, the horizontal axis, if we look at the pre-human and Maori period, you see there is some change on that axis, but not a lot. Whereas if we look at the European, we just see, a really large change, and that's happened in just 200 years. So often I look at this and I just wonder what we've done to our lakes in 200 short years. And what's really interesting about this pattern is that it's happening across our lakes, regardless of lake type. So this is the same data, but I've just separated it out into current trophic condition. And the different colours, the different shadings on those dots represent depth. So while the greatest changes is happening in the lakes which have become eutrophic or super eutrophic, we're also seeing change in our oligotrophic lakes. So some of our small alpine lakes are changing. And certainly we understand, or our interpretation of this is there's of course local scale drivers, so we understand them, intensification, invasive species, etc. But we really don't yet understand why our oligotrophic lakes, particularly some of those remote alpine lakes, are changing. And of course we can speculate about climate change, nutrient inputs for waterfowl, etc. And that's really a bit of an ongoing topic of research at the moment. Yeah, so to summarise the sort of biophysical section of my talk, it's really important. I think one of the most valuable things we've learnt was that every lake has its own unique history, and if we listen to the voices of our lakes and if we hear their stories, then we can better inform their management moving forward. We're really worried about this national scale loss of diversity and just what it means going forward in terms of resilience and resistance of our lakes. And again, just to highlight those local scale drivers which we understand, but those national scale ones which we still need to get our head around to protect some of our really precious alpine lakes in particular. Okay, so it was really a real privilege, I think, as part of this project to work with so many lakes, but equally rewarding was working with our Indigenous communities. And so I wanted to cover off a little bit of the work that we did in this space. So I'm going to focus on three key areas that we worked in, both relationship building, weaving knowledge streams, and science communication. So at the outset of the project, we set ourselves the mandate that we wouldn't sample a single lake until we'd had the appropriate conversations with the affiliated tribes, or as we call them in New Zealand, iwi. So this turned out to be 60 iwi across the country, and it probably took us the first year of the project to do this. So incredibly time-consuming, but also turned out to be one of the most rewarding parts of the project. And these images show the knowledge sharing mornings that we held in each region before we'd start sampling, and we'd take a sediment core and split it on site and have a look at some of the early changes that we would see. That we'd also hear from the iwi or tribes and learn about their long affiliations with the lake, and often that was an incredibly enriching experience. And what we learned that it was also incredibly important to maintain these relationships and to also return back and share the knowledge that we'd generated during the project. And that's something that often in our country doesn't happen regularly. Okay, so in 4 regions around the country, we developed much more in-depth relationships with iwi. We couldn't possibly do that across the whole country, so we focused on 4 regions. And one of the specific topics that we set out to achieve was how did we weave different knowledge systems? So how do we weave Indigenous knowledge with biophysical science? So I'm just going to show a short video which highlights one very small example of weaving knowledge. As that video goes, you're going to hear a couple of terms which are in Te Reo or Māori. So Mataranga Māori, which is Indigenous knowledge, and Te which means treasured.
Tēnā rākou te katoa, ko rewa ni rima hau, no te tai tokero, e mahi ana hau ki te whariwara o Victoria. There's a lot of talk about so-called Western science or conventional science and Mātauranga Māori. We should recognise that they are different worldviews, they have different methodologies, but those differences are telling us something as well. With science, it's all about objective observations, right? Detaching the personal from the target observations. Whereas those things are really important in Mātauranga, integral to the whole process, the connection of people with place, connection with the environment. But as I say, we can also find some overlaps. And I can give you an example from morphic pollen, like most science. It's not perfect, it's got limitations and it's got gaps. We can recognise plants in some cases down to species level, but in some cases we have to lump species together because the pollen grains are very similar between 2 different species. Take our flaxes, for example, in New Zealand. You know, the well-known harakeke, the common flax. The pollen grain is identical or very close in appearance to a related species called wharariki, which is a local species and more fitu, and which is actually a taona species with Ngāti Koata. So the Western science, if you like, the paleontology can only go so far in terms of telling us what flax is growing and when, and more fitu. But with the Mātauranga, we know what species of flax that is because Ngāti Koata have an intricate relationship with that species. And so we can put the label wharariki on the pomegranate that we see at Mawhitu. So there's an example of where the two knowledge streams, if you like, weave together and the one enhances the other.
Great. So another area that we focused on was science communication. So I've shown you lots of wiggly lines and dots, but we wanted to challenge ourselves to think about how we could better communicate our science with our Indigenous partners. And so there are a number of different ways that we did that, but one way we achieved this was by developing a virtual reality experience. So iwi or Māori are often storytellers, they're often very visual in how they like to see data presented. And so I'm just going to show you a little video of this. And so the sort of landscape and lake data was generated from a weaving of the biophysical knowledge, but also that Mātauranga Māori. And so this video starts in the present day, and we're looking, you can just see the landscape around the lake, and it's very devegetated. If we dive down under the water, you'll also see that it's a completely barren landscape. But if we were to travel back in time and go back 1,000 years, you'll see what an absolute gem of a place this lake is. It's beautiful forest right up to the edge of the lake. And when we dive under, you'll just see what a vegetated place it was and how full of life it is. We can also travel to the period where Māori settled and lived around this lake, and embedded in the lake embedded in the virtual reality experience are also touch points and videos which talk about the Mātauranga, the Indigenous knowledge of this lake and the biology and the cultural importance of this place. And really importantly, this virtual reality world includes Ngāti Koata, who are the iwi, their 100-year vision for the lake. I think this is incredibly important. This is a site of active restoration, but of course restoration of this lake is going to take many generations, and so much of the work that's happening now, is not going to come to fruition for many centuries, so it's really important to be able to see what the lake is going to look like in the future. Cool. And so I just wanted to finish up by sharing our adopted whakatauki, or proverb, for this project. Mi hoki whakamuri ki haere whakamoa, and that translates to current and future activities should be guided by lessons from the past. And while I leave you to reflect on that for a few minutes, I've just got a short video which shows you some of the adventures that we had as part of the Lakes 380 project. Voices whisper that I'm not to blame. They say everyone's afraid of change. Sounds to me like they've given up, but they don't know the heart I've got. I'm willing to change, I just don't know how. So I think we need to change that last line of the song to I'm willing to change and the time is right now. So thank you very much for listening, but yeah, more importantly, thank you very much for all the amazing work you do. It's so incredibly important. Thank you. And I think I've got a few minutes for questions.
Hi. Can you all hear me? My name is Amalia Handler. Thank you so much for this talk. I work for the U.S. Environmental Protection Agency, and part of my work is to support the National Aquatic Resource Survey Program, which includes surveys in the lower 48 states of 4 different water body types, and one of them is focused on the lakes assessment. So I really appreciate learning about New Zealand's, this project in New Zealand, and I'm going to be looking a lot more into the survey methods. I wonder if you could talk a little bit about how you decided which lakes to sample across the country.
Yeah, thank you very much. Great question. So it was a multi stage process. To start with, we wanted to cover a whole suite of different scientific gradients, and so we looked at size, land use, altitude, trophic status, but we also, through the consultation with our iwi partners, it was really important to also include culturally significant lakes, and there were a few lakes that were what we would call waihutapu, or sacred, that we were asked not to sample. So that became a really big part of the decision process, and also there was inclusion of some lakes which might have been had high social values, lakes that were part of targeted restoration programs. So a whole sort of multifaceted approach to coming up with the lakes. Yeah, access, a few other things that get thrown in there. Sometimes you'll see from those helicopters that it actually wasn't possible to land at the lakes that you'd aimed to get to. Yeah.
Thank you. I'm really impressed at the helicoptering into some of these sites.
Sounds fun, but you're moving a couple of tons of gear each time, and they need to be packed up and unpacked. So they were long, hard days, but yeah, amazing opportunity. Yeah.
Hi, thank you again for a wonderful talk. Can you talk more about, you said you were sampling across like a lot of scientific gradients for the lakes, and one of the things you noted was like the gray bands in the soil cores regarding like the storm events, and since those are expected to increase with climate change, do you expect any like specific scientific gradients to be affected by those storm events? I noticed that there was like a big gradient between certain lake types on the shore versus inland lakes, or oligotrophic or supertrophic. So can you talk more about like which specific types you're expecting to be affected? Sorry, that's where you...
Sure, yeah. So I guess we've got a few different interesting things there because we have 1,000 years of storm events, and during that period, of course, the catchments changed hugely from a catchment which was fully forested to one which is now in intense agriculture. And so if we look at response of the biological communities, at least our preliminary data, as you'd expect, shows it's much quicker and much and back to those original pre-storm communities when we've got an intact catchment and a healthy system. As we move into the more intensive agriculture, those systems are not going back to what they were like pre-storm events. So we've got a really interesting gradients within lakes. Then when you look across at a national scale, a lot of it in our country comes down to geology. And so actually that case study lake I showed you is in a place where the catchment is incredibly prone to erosion. And so we get the huge pulses of sediment, and the lakes are full of sediment for many months afterwards. And those are probably the main things. Catchment size plays we've got a really nice case study where we've got 3 lakes close to each other, but different sizes of catchments. So a large catchment